大家好，我们是第10组，以下是我们的组员。我们的标题是 Single Cell RNA sequencing reveals fibroblast heterogeneity and increased mesenchymal fibroblasts in human fibrotic skin disease。单细胞 RNA sequencing 揭示人类纤维化皮肤病中成纤维细胞、抑制性和增加的间充质成纤维细胞。成纤维细胞增殖和细胞外基质 ECM 过度积累的纤维化会导致发病和死亡，会影响任何器官，导致疤痕形成跟器官的功能障碍。至今，纤维化皮肤病的发病机制尚未完全阐明，仍然缺乏根治性的治疗方法。在这项研究中，对疤痕疙瘩克隆进行了 scRNA sequencing 分析。疤痕疙瘩是一种纤维化皮肤病的范例。疤痕疙瘩的成纤维细胞可分成4个亚群。与成纤维细胞相比 ，mesenchymal cell markers 的成纤维细胞显著增多。此外，成纤维细胞亚群可能是因为 postin 基因过度表达纤维化皮肤病的胶原蛋白。这些研究结果将帮助彻底了解纤维化皮肤病，并为纤维化治疗提供潜在的治疗方向。
To understand more, the human skin is composed of 3 main layers: epidermis, dermis, and subcutaneous. The epidermis gives rise to the hair and glands, contains keratinocytes and carnocytes which contribute to thermoregulation and barrier formation. The dermis provides structure, strength, and flexibility to the skin with proteins called elastin and collagen. It also contains other structures such as hair, glands, vessels, and nerves. Dermal fibroblast is the main cell type of the dermis. It has an important task in the healing of wound by populating the site of the wound to produce extracellular matrix, which forms a scar. Keloid and hypertrophic scars are examples of skin fibrosis. A scar is different from normal skin. It doesn't have hair, glands, and touch and pressure sensors. Recent studies say that fibroblasts are actually composed of different cell populations that differ morphologically and functionally, and that many cell types may contribute to fibrosis. But how does this heterogeneity emerge in developing fibrosis? So, single-cell RNA-seq was done to analyze cell heterogeneity and explore regulatory changes on keloid and normal scar dermis tissues. They get samples of keloid and normal scar tissues from patients with consent, remove the epidermis with DSpace 2, and dissociate or separated the dermis into single cells with collagenase 4. After filtering the resulting cell suspension, they sent the samples to Novogene for library preparation and sequencing. They utilized 10x genomics platform. They attach a specific barcode or unique molecular identifier to each cell. Cells were partitioned into the gel beads in emulsion or single-cell GEMs and proceeded with sequencing, data processing, analysis, and validation.
这里使用的是 UMAP Unsupervised Uniform Manifold Approximation and Projection Clustering 的方法，将所有 RNA 序列分群，总共分成21个细胞群，并且做 Hierarchical Clustering。然后将 RNA 序列中主要组成5个成纤维细胞群 C2、C8、C14、C15 和3个内皮细胞群 C1、C6、C7 提出做 Linear Specific Marker Genes 支系标记基因 COLE-A1 标记成纤维细胞支系 ENG 标记内皮细胞支系。在不同细胞比例的部分可以看到，克罗伊斑和疙瘩的成纤维细胞减少，而内皮细胞和平滑肌细胞则增加利于血管的生成。而在基因表达的不同，克罗伊的成纤维细胞基因表现程度最大差异。因为在纤维化过程中，成纤维细胞扮演着重要角色，再次使用 UMAP 的将成纤维细胞细分，总共分成13群。B 图中可以看到克罗伊和 normal scar 的分群差异，绿色的 SC4 分群增加。藉由前面所做的标记基因，可以将这13群分类成4个过去研究已知的亚群 Secretory Papillary secretory reticular, mesenchymal 和 pro-inflammatory。SC6、SC7 是 secretory papillary fibroblast，SC2、SC3、SC9 是 pro-inflammatory fibroblast，SC1、SC4 是 mesenchymal fibroblast，SC5 是 secretory reticular fibroblast。在 EF 图中可以看到克洛伊的 fibroblast 亚群中 secretory papillary、secretory reticular 和 pro-inflammatory 亚群皆下降，唯有 mesenchymal 亚群增加。据图呈现的克洛伊和 normal scar 的基因差异，其中有关骨骼系统的发育、骨化、成骨细胞分化相关基因在克洛伊的表现增加最多。接下来比较 mesenchymal fibroblast 在克洛伊和 normal scar 之间的差异，特别挑选了骨骼系统发育基因做比较，例如 COLCAE、COMP、POSTON， 利用 Gene Ontology、GSEA、Gene Set Enrichment Analysis 做分析，可以看到骨化、成骨细胞分化相关路径在 keloid mesenchymal fibroblast 中表现最多。
In terms of characteristics of mesenchymal fibroblast and fibrotic skin disease dermis, they found that skeletal system development ossification and osteoblast differentiation associated genes such as POST-N and COL11A1 were enriched in mesenchymal subpopulation. The heat map in figure B suggested that gene expression was significantly different between the mesenchymal subpopulation and other subpopulations. Here in figure C, the increased genes in the mesenchymal subpopulation included some secretory proteins such as POSTN, COMP, COL11A1, COL12A1, and COL5A2, and some other membrane proteins such as STC1, ADAM12, and CD266, while CD9 was decreased in mesenchymal subpopulation. The figure D shows the functional enrichment of upregulated genes in mesenchymal fibroblasts. Here in figure E, violin plots showing the master regulators for mesenchymal fibroblast in the right panel. To validate the findings identified by scRNA sequence, here they performed immunofluorescence staining for NREP and ADAM12 in normal keloid tissues. In terms of potential ligand receptor interaction analysis in mesenchymal fibroblast subpopulation, in normal scar, interactions between secretory reticular fibroblasts and other cells were most evident, but in keloid, interactions between mesenchymal fibroblasts and other cells were most evident, suggesting the important role of mesenchymal fibroblasts in keloid development. These ligand receptor circles indicated that fibrosis signaling interactions such as TGF-beta, POST-N, and PDGF were significantly increased in keloid as shown in D compared to the normal scar as shown in C. Here, they isolated CD266 plus or CD9 minus and other fibroblasts from keloid dermis by flow cytometry as shown here. QRT-PCR and Western blot validated that mesenchymal fibroblasts maker genes were significantly enriched in CD266 plus or CD9 minus fibroblasts compared to other fibroblasts as shown in figure B and C respectively. Here, GO and GSEA analysis showed that upregulated genes in CD266 plus or CD9 minus fibroblasts were associated with extracellular matrix organization, skeletal system development, and so on, indicating these fibroblasts had the mesenchymal fibroblast identity.
The mesenchymal fibroblast subpopulation included subgroups SC1 and 4. The diffusion pseudotime DPT analysis arranged them into a major trajectory or directions. It estimates the temporal order of differentiating cell in single-cell RNA-seq data. Fibroblasts from SC1 and 4 were distributed across the pseudotime space, with SC1 cells occupy the lower space of the major trajectory and SC4 cells occupy the upper space. Also, the mesenchymal progenitor marker genes, such as post-N and col11A, occupy the upper space. RNA velocity analysis distinguished 4 sets of vectors defined as paths. From the paths, we can see a branch trajectory with 2 major branches, path 2 and path 3, 4, and a pre-branch path 1, which represents the initial states of fibroblasts. SC4 fibroblasts compose the vast majority of the pre-branch path 1 and path 2. And SC1 fibroblasts compose the majority of path 3 and path 4. This analysis suggested that SC4 may be lower differentiated than SC1 fibroblast. The difference in expression levels are shown. Gene ontology and GSEA analysis suggested that extracellular matrix organization, ossification, chondrocyte development, and so on were enriched in SC4 fibroblasts compared to SC1 fibroblasts. In summary, these results indicate that SC4 fibroblasts are more mesenchymal-like and may be more important in keloid development. They compared fibroblast scRNA-seq data from a recent scleroderma research with their number 3 normal scar fibroblast data because they had similar number of fibroblasts. They performed unsupervised clustering and observed heterogeneity with 9 subclusters. Cluster 7 was increased in scleroderma compared to normal scar. Mesenchymal fibroblast markers post-N, ADAM12, COMP, and NREP were extensively expressed in scleroderma cluster 7 fibroblast. Correlation analysis suggested that scleroderma cluster 7 fibroblasts were most analogous to mesenchymal fibroblasts in keloid with 0.962 correlation value. Immunofluorescence staining results showed that the proportion of ADAM12 plus NREP plus cells was higher in scleroderma tissue than in normal control tissue. In summary, these results indicated that increasing mesenchymal fibroblasts may be a universal mechanism in fibrotic skin diseases because it was also observed in scleroderma.
最后做个统整讨论，从 scRNA sequencing 定序中可以分出13个 fibroblast 细胞群。Fibroblast 细胞群又可以分成4大亚群。Mesenchymal 亚群在克洛伊疾病中明显增加，并且被认为是皮肤纤维化的共通特性。Mesenchymal 细胞群中的增加，其中一个特性是分泌细胞蛋白的增加。细胞的纤维化疾病，相较于正常伤疤的细胞交互作用，集中于 mesenchymal fibroblast 对其他的细胞。对于 fibroblast 的子群 SC4， 在纤维化皮肤中会大量的表现。SC4 细胞中成骨和软骨形成相关分泌蛋白会表现较多。SC4 可分化成 SC1 成纤维细胞。总结，整篇论文是在讲述一个以单一细胞为单位研究 fibroblast 抑制性，并且发现纤维化疾病中 mesenchymal fibroblast 的增加与参与胶原蛋白的过度表达。研究结果有助于寻找纤维化皮肤疾病的致病原因以及治疗标靶。